This is Chapter 18, Section 2, and it's on viral structure and reproduction. Code word pizza. The key concept here is that viruses exist in a variety of shapes and sizes. So a virus. Viruses, so whenever you get sick, you can either have a viral infection or a bacterial infection. Bacteria we've talked about. Bacteria are your prokaryotes, which were the cells that they have no nucleus and they have no organelles, but they're still cells. They're still, we would call them organisms. And when we talked about the different levels of life, the different classifications of life, um, they fit into life. Whereas viruses um, are actually not considered living things, depending on who you talk to. And that's because a virus, all it is just a protein coat in DNA. A virus cannot exist on its own, whereas bacteria can exist on their own. They can have, they can do their thing. They don't need to infect you. They can live on their own. Whereas a virus has to have a host. Um, so, getting into the different shapes and how a virus enters a host, first thing we'll talk about is the shape and the structure of a virus. As I said, it's a very simple structure where you have the genetic material, where the genetic material for the virus can be, it can be DNA, or viruses could just have RNA. It depends on what kind of virus it is. Some are made of DNA, some are made of RNA. To remind you the difference of DNA and RNA, remember DNA was the ATCG, and whereas RNA had the U instead of the A. Other than that, DNA and RNA are very similar. DNA is double-stranded. It's two-stranded, and it's that helix, whereas RNA was single-stranded. RNA is what we use to make proteins in bacteria, and in prokaryotic cells and eukaryotic cells. You went from DNA, the main copy, to RNA, and then from RNA you made the protein. Um, so. But for viruses, how they contain their genetic material, how they contain their instructions to make proteins, can either be in their DNA or they could just have RNA. Okay, so they have their genetic material, their nucleic acid, and then they have what's called a capsid. And a capsid is just a protein shell. Okay, it's just it's basically like it's like a shield for it. You can think of the capsid as just this little protein shield that protects the DNA. It's just kind of like a storage container for the DNA. Some viruses can have another layer, another outer coat called a lipid envelope. So these two bacteria or these two viruses right here have a lipid envelope. It's what's in yellow. Um, and then whereas this bacteria on the far right only has the capsid. Examples of envelope bacteria are your influenza, which is your flu virus. And when somebody says they have the flu, that means they got infected with a virus. They got infected with this virus here specifically, the influenza virus, and it looks like this. Um, Rabies is it's what's called a helical virus, where the DNA is wrapped in this little helix here. Looks like this. And it's kind of got this rod shape to it. Rabies is you hear about that with like dogs, or wild animals. You'd worry about if you see an animal in the wild is, oh, they say that animal might have rabies, and what they're referring to is they might be carrying this virus, and if they bite you or something, that they could transmit this virus to you. Anyways, that's what this virus looks like. And then there's a polyhedral virus where it looks like almost like one of those geometry shapes where it's got these all these little sides, so, like a polyhedron or some sort of octagon or something. Okay, moving on. Then there's a type of bacteria or type A of virus called a bacteriophage. A bacteriophage is just a virus that infects bacteria. And kind of the name of it sort of gives it away, where it has bacteria literally in the definition. And a bacteriophage has this, they almost look like a syringe or like a, I don't know, they kind of got these little legs. I mean, this is literally, what they look like. They have these little legs, and then they have this tail. And these little legs, they call them tail fibers. And this part right here, they call the tail sheath. And then they have the capsid, which again, the capsid is that protein coat that's like, you could think of it like a, almost like a storage container, where on the inside of that storage container you have the nucleic acid or the DNA. And again, this DNA could also be RNA rather than DNA. It could be one of the two. Okay, so a bacteriophage can get into a host cell in different ways. And we'll go over these different ways. So this right here is in yellow here. This is a bacteria. This is an actual picture, a scanning electron micrograph, an actual picture of a bacteria with its shape. And then these right here are your viruses. So it kind of gives you an idea of just how small these viruses are. There's hundreds of them on this one bacteria, and they are attaching themselves. This is a zoomed in picture of it. So there's those little feet of it where it attaches to it, and what it does is it injects its DNA. Think of it like a syringe, like like a needle in a syringe where where this bacteria literally will stick almost like a needle into the bacteria, and it will just inject its DNA inside the bacteria. Okay, so how a virus can get into whether, this is any type of virus, not just the bacteriophage, but any sort of virus. How it can get into the cell is by endocytosis. Endocytosis, and this is especially for eukaryotic cells, for viruses that infect eukaryotes, which, to remind you, eukaryotes, remember the EU. When you see EU, you think you, as in you are a eukaryote. They enter by endocytosis, and endocytosis requires energy, which means it's a form of active transport. Those are all concepts that we covered in the fall. There's also passive transport. Passive transport is where things get into the cell. This right here is a cell. Inside of the cell, I'll say that with an I. Outside of the cell, I'll say that with an O. Where if you go from outside of the cell to inside of the cell, if it was passive transport, these molecules out here go from outside the cell to inside with no energy required. Whereas active transport is when, in order to get these blue spheres to go from outside the cell to inside the cell, you have to use energy. So viruses get into the cell through endocytosis. So endocytosis is where you use energy to make these little vesicles, these little pockets that form in the cell membrane. This is the cell membrane, the outside of the cell, and you form these little vesicles. So each one of these little blue dots would represent a virus. So these are all viruses out here where they would approach the eukaryotic cell. So one of one of your cells or another animal cell or a plant cell, and they form these little vesicles, and then they come into the cell. Another way a virus can get into the eukaryotic cells is by fusing with the membrane. So rather than the membrane making these little vesicles, rather than making these little vesicles like in the other slide, the virus would, this would be the virus right here. It would come to the cell, and then it would just fuse with the cell membrane, and then dump its contents into the cell, like so. Code word sweatpants. Okay, so there's two types of viral infections. There's what's called a lytic infection. And a lytic infection is going to cause the host cell to burst, or literally cause the host cell to explode. So these are the steps to the lytic infection. And how they drew it, they showed it for a bacteriophage, which remember bacteriophage is one of the viruses that infects a bacteria. And they remember they're almost like they're like a needle and a syringe where they inject their DNA. This is that or RNA. This is the genetic material of that virus that they inject into the cell. So it attaches to the cell and then it injects its DNA. And then that viral DNA or RNA, the viral genetic material, is going to form a circle. And then from there, this circle, this has all of the instructions for the proteins, all the instructions needed to make another virus, all the proteins that are needed to make another virus. So this next step, because the virus doesn't have all of the different machinery, all the different parts that normal cells have, the virus essentially will use the machinery of whatever cell it infects to make new viral parts. So it's going to use the ribosomes and the endoplasmic reticulum and the Golgi apparatus, all those different parts of the cell that we need to make and process proteins. The virus is just going to use the host cell's machinery. So it uses the host cell machinery to make its new viral parts. So this is, it made all those new viruses. And then the last step, the virus just, it's just a jailbreak. It just explodes. They're trying to show this cell exploding. And then these viruses just explode on out of the cell. And then they go and travel on to other areas of the body and infect other cells. So whenever you feel like a sore throat and your throat's all scratchy or uncomfortable whenever you're sick, what's causing that is kind of in part because you have viruses that are infecting the cells in your throat, and they're literally exploding some of the cells that make up your throat, and that's it's part of what makes your throat feel all weird and crappy and all right. Anyways, that's the first one, the lytic infection, and the second one is called a lysogenic infection. For filling in your video notes, you're going to need to look at these steps here. But to explain the lysogenic infection, I'm going to jump over to the next slide where it compares lytic versus lysogenic infection, because I think that might explain it a little bit better for you. So I'm just going to go ahead and jump to the last slide. Code word Netflix. Okay, there's a lot going on in this slide, but just kind of stick with me. So up top here, you got your virus. This one is, again, one of the bacteriophages, where it's got the capsid, that outer protein coat, and then you have the DNA. The virus is going to attach to the cell, and it's going to inject its DNA, represented by this. Okay? Then it's going to form a circle. And then from here, it's got two paths. It can either become the lytic cycle, the lytic infection, where it uses the host cell to make other viruses and then explode on out, or it can go into a lysogenic cycle, where the lysogenic cycle is the bacteria, rather than going all out and making new viruses, the virus chooses to basically lay in wait. It essentially goes into hiding, and it does that by combining with the host cell DNA. So the virus joins up with the host cell DNA, which is represented by that, and it becomes what's called a prophage. And then, whenever the host cell is doing mitosis and it's dividing and making new cells, the viral DNA is combined with the host cell DNA, and this could go on for years. It could go on for quite a while. The host cell could just keep dividing and dividing, and years could go on by, and just keeps dividing, and then all of a sudden the virus will just choose, hey, I'm gonna come on out. I'm gonna come out of the closet, if you will. I'm gonna come on out of this host cell DNA, and I'm gonna turn into this viral DNA, and I'm gonna go into the lytic cycle where I start making new viruses, and then I explode on out of the cell. So, example of a lysogenic infection. So, why a virus might want to go into the lysogenic cycle is maybe the virus doesn't want to explode on out of the cell for whatever reason. Maybe they just kind of want to hang on out into the body and lay in wait, and before they kind of make their move, so to speak. Um, you, the HIV virus. HIV is what causes AIDS. So people who have AIDS, they have what's called the HIV virus, or the HIV virus undergoes the lysogenic cycle, where it combines with the host cell DNA, and it just lays in waits until after a certain period of time, sometimes it's many, many years, it'll eventually come on out and it'll start making new viruses and explode on out of the host cells. Um, and that's what happened, and that's why people who have AIDS, a lot of times they'll have the HIV virus, and they won't know it because they don't have any symptoms for many, many years until that virus decides to go into the lytic cycle to come out of hiding, so to speak, and start making new viruses. Where it starts, turning into AIDS, or they start having their immune system declining because the immune system's trying to keep up with all these viruses that are invading the body and killing off all their immune cells. Okay, that'll do it for this video.